thank you very much for inviting me to come and talk to you today. I'm going to talk to you about a project that is a work in progress still. I've been doing it for about 3 years now. Sorry, I seem to be echoing. I've been working on the NERC-BESS project, which is Biodiversity and Ecosystem Services and Sustainability, which is not terribly catchy, but I've been working on the Wessex BESS project, which is based on Salisbury Plain. They're interested in how ecosystem services are provided on chalk grassland, chalk streams, pollinators, and so on. So my remit is to try and disentangle how plant species diversity on the chalk grasslands contribute to carbon sequestration and how we can optimize species choice and try and speed up restoration. I seem to be incredibly echoey, sorry. So I've been working with all these people. Sadly, Nick, one of my bosses, can't be here today. So my background is in plant functional effects traits. And I guess most of you are probably geneticists and you have probably a different definition for traits. But my definition of effects traits are characteristics of the plant species that will directly impact its surroundings in terms of ecosystem functioning, such as carbon flux and lockdown and nutrient cycling. Things like tissue nitrogen concentration and leaf thickness and so on. And a number of years ago now, it sort of started to generate a lot of interest to see how we can move away from taxonomy and into a more sort of functional definition of how plants are actually affecting their surroundings. And it kind of gave rise to this leaf economic spectrum, which kind of simply divides plant characteristics into very fast processing and very slow processing, in its most basic definition. So this diagram will show you that the faster ones have higher photosynthetic rates, higher soil respiration rates, and root respiration rates. They might have more nutritious leaves, which is more appealing for decomposers or herbivores, so it speeds everything through the system quite quickly. On the opposite side, you've got everything that moves very slowly and very carbon-rich organs that don't really decompose that quickly, and everything is quite slow. So from this, you can derive simple hypotheses. This is a really basic one that I just to illustrate my point that if you've got high nutrition inputs, then you might have a faster and more bacterial microbial community that processes things very fast. But that might also give rise to a lot of losses and less storage of carbon and so on, because it might be being sort of metabolized quite quickly. Big problem with root trait science is that, especially when you're working on a landscape scale or a big plot scale, you have this big grassland with loads of different species. It's almost impossible to characterize what the roots are doing just by looking at it. And if you've got a long-term study where you want to come back season on season, you can't just dig up your plants and go, oh, okay, your roots are doing this and that. And there's the probability that you're actually going to lose a whole bunch of roots. So they've kind of been overlooked in a big way. And also the pressures on roots are different to the pressures on shoots. The herbivory is different, the spatial requirements are different, soil depth and so on, as opposed to almost infinity above ground. So it's very hard to know what's going on. And as a result, these plant trait databases that have arisen in the last 10 years or so, you see this top right-hand corner, that is the TRI database. It's got over 1 million entries of traits, different species from around the world, all sorts of different traits. That tiny red sliver is the roots. It's 0.8% of over 1 million entries is root traits. So you see the problem. And it's led to calls from Richard Bargett, my boss, and various others saying, we need to get a handle on this. We need to figure out how we can use roots, because they're the main driver of almost every process that we're interested in. And just as just to illustrate my point further, This is an entry from Ecoflora, which is quite a big important traits database. Arabidopsis thaliana, arguably one of the most studied species in the world, and it doesn't even have a full complement of data in this data set. So you see the problem. So I had a bit of a think, because I was given this chalk grassland, and I was told to create an experiment to try and unpick carbon sequestration and which are the best species to use and the best mixtures and complementarity and so on. And I had a bit of a think, and I decided that maybe it's best to come up with some really simple, as universal as possible traits. And that's not easy to come by. There's a lot of people working on roots and exudation and so on, and it's an incredibly complex field of study. And the associated microbes and so on. I mean, we've heard from a number of people this morning about associated rhizosphere microbes and so on, and it's incredibly difficult. And from my point of view, it's too difficult for a landscape scale study. And if I want to disseminate this to farmers, and if I want to if restoration and so on, I need to come up with something simple and snappy and easy. And so what I came up with is root depth class, not a continuous variable, but shallow, medium, and deep, and basic architectural classifications. So I realize it's not a perfect classification, but if roots basically go in complexity in my chalk grassland, I'm not saying this is true for everything, but they go from tap roots, very simple, sort of carrots and so on, and you have wild carrot on the chalk, all the way up to these incredibly complex fibrous roots. And in the middle, you have rhizomes that move laterally across the soil and have their own functional footprint that's starting to be recognized now. And I thought that might be the simplest way of doing it, is categorizing all the species that I have potentially in this landscape, and categorizing them by these two different traits. And I also threw in things like perenniality, so how long they live, which is also not a direct measure of how fast things cycle, but it helps. And tissue thickness and a couple of other things. Roots are incredibly important, though, and so I weighted my classification by the roots, because they are the main contributor of carbon to the soil. And whether you have shallow roots, then the carbon's going to be disseminated into the top layer. If you've got a really deep one, then it's going to be put into the bottom, the very deep layers. And very simple hypotheses here. If you're putting carbon very deeply into the soil, then it's likely that it's less available for microbial activity because it's more anoxic. It might be slightly more waterlogged, and so on. So it might be more stable and less prone to metabolism by microbes if you put it deeper into the soil. Likewise, with my root architecture, you see the second one from the left. That's obviously a fairly fibrous root. There's lots of opportunity there for microbes to take hold, to collect exudation, carbon sources and so on, compared to the third one, which has a very low surface area to volume. And so, simple hypotheses, because a chalk grassland is an incredibly complex thing. You can get up to 40 species per square meter. And trying to characterize a chalk grassland based on plant characteristics is actually really difficult. There's some evidence that my little theories are backed up, sorry. There's somebody called Tina Roos who works at University of Southampton, and she did a study in 2008 that shows that the zero order roots, and that's that one there, that's the initial tap that goes down, picks up less nutrients from the soil than the second order, which are the first laterals. And so it goes on. As you get more laterals, you get more nutrient uptake. And so that kind of supports my theory a little bit. So I'll get on to my big study in a minute, but I was a bit concerned that this might be a bit much for potential reviewers when I come to publish the actual study. So I decided to do a smaller experiment in the greenhouse to see if any of my ideas actually held water, if it worked. And the key thing that I was interested in was plasticity with this one. And I just wanted to see if, as roots become more complex, do they also become more plastic? So can they shift to collect resources in different patches, or water if it ends up in a certain location, and so on. And that would, that would compound my ideas on, this root architecture that, I've kind of put all of my eggs in that basket, as it were. And so what I did was I grew a whole bunch of plants in the greenhouse, and they were in these sort of 4 basic classifications: fibrous on the left, rhizome, stolon, and tap. And you can see that they're sort of getting less complex as they go up. And what I just wanted to see was, if you apply water in different places, in the top of the column or in the bottom of the column, if you get a shift in rooting biomass. And the answer is yes, you do. So the well-watered, what we've got here are the columns in this. The one on the left is the very top 5 centimeters, and then they go down in 5 centimeter increments as you go on. So what you would normally expect to see is most of the roots would sit in, around the top, sort of 5 to 10 centimeters, and then you get less as they go down. That should be what you expect. And that is what you get. So we have fibrous, rhizome, stolon, and tap. And so when you put the water in the top, not a great deal happens. But when you put the water in the bottom, you see this shift of allocation of biomass. I'm not going to use the pointer because I get confused. You get a shift, and everything becomes a lot more even throughout the column, so the biomass is pretty much evenly distributed as it's trying to get to the water in the bottom, except for the taproot, which is that one on the far right. And it would appear that that is completely stuck. Once it's put its tap in, it's run out of resources to shift its strategy to slough off a bunch of roots and change where it's going to go. So I thought that was quite useful, and it does validate my bigger study. So Salisbury Plain, it's subject to a huge number of pressures. It's our biggest area of chalk grassland, I think, in the entirety of Europe, possibly, certainly for the UK. And there's all sorts of people that are trying to stake a claim on it. There's tourism, there's building, there's the MOD, the army, and so on. And so that we're trying to protect what is remains, and build up the fragments. It's a problem, though. It doesn't do itself any favors because it's so poor. The soil has virtually no nutrition in it at all, and it's about that deep. It's about 10 centimeters deep in the species-rich areas, and everything takes years, absolutely years, to grow. It really, all the true chalk species fall onto the left-hand side of this leaf economic traits, the really slow processing plants, and they can take sort of, a few years to come to maturity, whereas in normal grassland it'll be, a growing season or 2. So to restore a chalk grassland from an arable field, which is what a good deal of it has been converted into, back to the species-rich chalk grassland will take a century. And it's partly because the overloading with nutrition and so on is you can't really remove it, I suppose. You just have to kind of wait for it to disappear. So I was given an area of chalk grassland, and I was told to create a factorial study with different plots, with different functional groups. And so I decided that I would use the database data that I could find with the root depth data classes and the root architecture, perenniality, and so on, and use a cluster analysis on the species that you find in these areas to create 3 different functional groups. And what I found was that they do cluster quite nicely. I mean, it's not perfect, of course, but what you get is, on the left-hand side, you get mainly tap-rooted species, or big, thick roots, at any rate, quite simple roots, quite a variable longevity. When most of the species are actually incredibly long-lived, this group 1 actually is quite short-lived on the whole, and quite thin, productive leaves. In the middle, I have my tiny little species, lots of herbs, lots of Lamiaceae, things like prunella, self-heal, and clinopodium, cowslip. They've got these tiny little roots and little rosettes, and they don't seem to exert a great deal of influence, I suppose, on the surroundings because they're so small and sparse. And then on the right is my group 3 species. Quite a few of the legumes fell into that group, but there's quite a few in number 1 as well, actually. But they're the fibrous rooted ones, the ones where they all sit on the top of the soil. So straight away I can start to think, well, maybe those are the ones where they're going to be processing more because all the roots are sitting in the top. Those are the potentially plastic ones, and so we might have more higher process rates with those. And so we grew them in 2013, and we started to take ecosystem function measures. And here's one example. Group 3, when they're present, you get much higher rates of both ecosystem respiration, so that's soil and root respiration. Throughout the season, just across the board, you get very, very high respiration and also higher photosynthesis when these species are present. So that's not really conducive to carbon lockdown because a lot of it's being lost from the soil surface. So this photosynthesis, it's basically a measure of carbon drawdown, so that's why it's negative. But yeah, so when these fibrous species are present, you get very high flux rates. You also get higher soil nutrient values. So this is potassium chloride extractable ammonium on the left, and mineralization rate, which is microbial provision of nitrogen in plant available forms. And again, the fibrous roots, you get higher rates of that. So I suspect it's a more wasteful group. It's not really provisioning much, but then on the other hand, there's a lot of potential soil formation happening. So last year we put up rain shelters because we were interested in resistance and resilience. So we had there's been some studies that show that you really have to have a roofed control because you get these strange shading effects, particularly on the chalk. And hardly anything happened. And I think it's because on chalk, my soil is literally that deep. 10 centimeters is virtually nothing, and it's so incredibly fine and sandy. There's virtually no organic matter at all. And a lot of my plants grew with hair on when they shouldn't have hair, which doesn't help matters. And I think that they're so stressed in the first place that this, so you can see drought, like this is just a list of my ecosystem function measures that I took. I think they're just so stressed in the first place that hardly anything happened because they were expecting it. So, there's a few bits and pieces, but it's not a consistent thing. And this is what happened in Buxton as well on the long-term study that they have up there. But there were some funny shading effects. The roofed control actually had stronger effects than the drought in a lot of ways. And this is one interesting relationship. And it's been found a number of times, actually, that shading has stronger effects. And I'm not quite sure why, if it's some kind of photosynthetic reduction. But this is just root ingrowth and just scaled up across the plot. And so when you have these fibrous roots, they're always smaller, but for some reason when you have taps and rosettes on their own in the plot, they're actually better off under this roof than when they're well watered, when they're unroofed altogether. So this is fairly initial data, and I'm a bit confused because this has happened in a number of different things, and a number of different functions. Decomposition shows a similar kind of pattern, and I think soil ammoniums shows the same pattern as well. So it's a bit strange, and it's something that I'm going to need to pick through and try and add some more covariates to, I think. So I've come up with one potential question why that could be. So then I calculated resistance metrics, and if you get a value of 1, it means there's absolutely no change compared to the control. If you get a value of 0, it's 100% change from the control. And if you get negative values, it means that your drought or your stress has overshot the well-watered or the control. And that can happen if you get a microbial flush after you alleviate drought. And that's quite a common effect. So you'll suddenly get a huge spike in respiration, mineralization, and soil nitrogen and so on. So I have another interesting effect where if you have no rosettes and no taps, you get almost complete resistance. So virtually nothing happens. So that means that there's only fibrous, shallow-rooted species present in those plots, and I have almost total resistance. If I have both taps and rosettes, again, I get very high levels of resistance. But if one of those is missing, then my resistance completely hits the floor, and everything is incredibly unhappy. And I've just shown you the soil ammonium, because that seems to be quite an interesting one. It's given me results consistently. But you also see it in soil phosphate levels, in decomposition rates, respiration rates. So this is obviously something that's quite a robust result. There's something about these fibrous-rooted species that can resist drought, whereas the other species seem to need each other. There's some kind of facilitation, and it could be something to do with water, hydraulic lift and so on, which has been seen in these species before. And going back to my traits, my functional traits, and moving away from just simple group classifications, I calculated the actual plot level mean of the traits in my different plots. And just to show you that one thing that you do find, mineralization rate, you find that you need a couple of different traits. One trait is not enough to predict a given variable just on a plot level. It is enough for a landscape scale or a countrywide scale. One trait will quite often be enough. But at a plot level, you need a few, or one or two. And so in this case, what I found is that mineralization rate is quite strongly driven by how tall the plants are. So I think my little rosette species are actually driving this in a big way, and also how deep the roots are. And I've just gone for shallow, medium, deep. That's just a simple class. But it seems to be that mineralization rate is higher in very small species or deep-rooted species. And if you've got a mixture of both in your plots, then you're doing even better. I was going to talk about resilience, but it's even more complex, and I've probably run out of time. So I'm just going to wrap up with these philosophical thoughts, which are that chalk is pretty resistant to drought in general. There's not a great deal going on. It's not like runaway plant death or anything like that. And I think it's because they're so shallow. But that's good news for chalk grasslands, because they're so threatened in every other way. And also the functional effects traits are incredibly useful for, even in this incredibly coarse way that I've employed them just for this experiment, they're actually really, really useful for predicting function. So thank you very much for putting up with me. This is the MOD site on Salisbury Plain. And thank you to everyone that helped me.